So ladies and gentlemen, we are all welcome to my YouTube channel. Today we are going to look at the digestion and absorption of protein. So today we are going to look at the digestion and absorption of protein. So let's start with the word digestion. What is digestion? Digestion is a breaking down of large insoluble food molecules into smaller ones for the absorption of the animal. So when a large molecules of food is being broken down in smaller one for the absorption of animal is called digestion. It's called digestion. So large when large molecules of foods when large molecules of foods are break down into smaller ones for the absorption of animal, the process is called digestion. So generally, when we take food material, they are generally large, and the large molecules cannot be taken or cannot be absorbed by the body. And it can also not be utilized by the body. So you need to break it down from this large form into smaller ones which can be easily absorbed by the cells. So the process in which large molecules of food are being breaking down into smaller ones is called digestion. And these digestions, we have different types of food molecules. We have digestion of carbohydrates, we have digestion of protein, and we also have digestion of lipids. So ladies and gentlemen, you can visit my video on digestion of carbohydrates for the digestion of carbohydrates. But specifically in this video, we are going to look at the digestion and absorption of proteins. So what is a protein? A protein is a polymer, it's a valuable polymer of amino acids. A protein is a biopolymer, of amino acids. So protein is a biopolymer of amino acids. It means that so many amino acids are joined together to produce a protein. So it means that an amino acid is made of hundreds, 20, 30, and 50, and thousands of amino acids. So it means that proteins are made of amino acids. It means that the building block of proteins is an amino acid. And these amino acids are linked together by what we call peptide bond. So each amino acid joined with another amino acid by what we call peptide bond. So each amino acid linked with another amino acid with what we call peptide bonds. So the bonds that link each of the amino acids is called peptides. It's called peptide bonds. So each amino acid linked with another amino acid using peptide bonds. So that is how they all linked with one another. Peptides bonds. So the bonds that link one amino acid to another is called peptide bonds. So the protein, the way it is too large. It is too large to pass the membrane of the cell. So the way the protein is, it is too large to pass through the membrane of the cell. So if it is large to pass through the cell membrane, it means that the protein cannot get into the cell membrane. So the solution is that the protein must be digested. It must be digested into amino acids. So the process in which a protein is digested to produce individual amino acids is called hydrolysis. It's called hydrolysis. So the process by which a protein is breaking down into individual amino acids, of course, is digestion. But through the process called hydrolysis, using a group of enzymes called hydrolases. So hydrolases are the group of enzymes that involve in the breaking down of the peptide bond in the amino acids into the individual, breaking down of the peptide bonds in the proteins to produce individual amino acids. So the process of doing that is called hydrolysis. So digestion is usually occur using the process of hydrolysis, using a group of enzymes called hydrolases. So the process in which proteins are being break down through the breakage of the peptide bond using a group of hydrolases enzymes into an individual amino acid is called protein digestion. It's called protein digestion. And the group of enzymes that does that is called hydrolases. So now, generally in the digestion process, this group of enzymes, hydrolases, this group of enzymes called hydrolases, they are usually produced in their inactive form. They are produced in their inactive form. So generally these hydrolases, they are a group of enzymes that are produced in the inactive form. Why they actually provide through the inactive form is because to prevent the self-digestion. Because generally our body is made up of protein, and of course if we have these enzymes in their active form, or this group of hydrolases in their active form, they will end up digesting our protein. So to prevent self-digestion, so these enzymes are generally produced in their inactive form. They are generally produced in their inactive form so that to prevent self-digestion. So the reason why they are produced in their inactive form is to prevent self-digestion. Is to prevent self-digestion. So that is why most of these group of hydrolases enzymes that are produced in their inactive form. So first we said proteins or digestion is a process in which a large molecules of food are being broken down into smaller ones for the absorption of the body. So it's called digestion, and specifically we said that we are going to look at the digestion of proteins, and we said that proteins is a biopolymer of amino acids. It means that proteins is made of thousands, and it's made of hundreds and thousands of amino acids, and these amino acids in proteins are linked through a peptide bond. So to digest the proteins, the peptide bond must be break down using a group of enzymes called hydrolases. The process is called hydrolysis. So this digested, or when this peptide bond is being broken down and individual amino acids are produced, they are produced for the absorption of the animal. As we said that proteins cannot be absorbed or cannot pass through the cell membrane. So if any nutrient cannot pass into the cell membrane, it means that it is of no use to the cell. So it must be digested. It must be digested into smaller ones so that it can pass into the cell for the cell to utilize. So it must pass across the cell membrane. So now, most of these enzymes that are produced, they are hydrolysis and they are producing the inactive form so that they prevent self-digestion. So the general name given to the group of enzymes that digest proteins is called proteases. So this is our protein. So the general name given to the enzymes that break down the protein into amino acids, into individual amino acids, is called peptidases. It's called or proteases, proteases, or you can even call it peptides. You can even call it peptides. So this is the name given to the general group of enzymes that digest proteins. And of course, these peptidases are divided into two. We have two types of peptidases. We have number one, we have endopeptidase. And then the second one is exopeptidase. So these are the two groups of peptidase enzymes that digest proteins. And of course, when we said endopeptidases, so endopeptidase are those class of peptidases that cuts the peptide bonds in the protein. They cuts the peptide bond inside the core of the proteins. They actually break down the peptide bond from inside the core of the protein. Inside the core of the protein, produce small fragments of peptides. We produce a smaller fragment of peptide. It means that the endopeptidase do not completely break down the proteins into the individual amino acids. Rather, it produces some smaller fragment of the peptides. And the enzyme that does that is called endopeptidase. So it does it from inside. And we have exopeptidase. And what this exopeptidase does is actually it breaks the amino acids from amino terminal or carboxyl terminal of the amino acids. It breaks down protein from amino acids, or carboxylic terminal. So the exopeptidases break down the amino terminal or carboxyl terminal of the protein, and that is what they did. An example of these exopeptidases, we have aminopeptidase, and then we have what we call carboxyl peptidase. We have carboxyl peptidase. And examples of these carboxyl peptidase, we have carboxyl peptidase A and carboxyl peptidase B. So these are the exopeptidases that break down the protein from the amino acid or carboxyl terminal, and it's divided into two. We have amino peptidase and we have carboxyl peptidase. So as we said from here, it's generally proteins are being broken down into amino acids, and the name given to the general enzymes that those work is called proteases or peptidase. And these peptidases are divided into two. We have endopeptidase and we have carboxyl peptidase. So endopeptidase is breaking the amino acid inside the core of the protein to produce a smaller fragment of the peptides. And then the ectopeptidase is usually break down the protein from the amino acid or carboxyl terminal. So that is ectopeptidase and endopeptidase. And as I said, there is also so many group of enzymes that are produced in the inactive form. There are so many of them. The name given to the enzymes that are produced in the inactive form is called proenzyme or zymogen. So whenever you produce enzymes and it is inactive form, the name given to that enzyme is called proenzymes or zymogen. So now we are going to look deeply, or we are going to look at this protein digestion deeply. So now we are going to look at properly the digestion of protein. So generally, when we take dietary protein, the daily requirement of dietary protein is between 50 to 100. It's between 50 to 100. So the daily intake of dietary protein is usually between 50 to 100 gram per week. That is the daily protein intake. The daily protein intake is usually between 50 to 100 gram per day. So when dietary proteins is being taken, so the first thing is the action of what we call gastric mucosa. So when you take a dietary protein, a gastric mucosa stimulates the production of a hormone called gastrin. So gastrin is a hormone. And of course, ladies and gentlemen, we should know that the digestion of proteins do not take place in the mouth because the enzymes and the environment is not actually favorable for the digestion of protein in the mouth. So therefore, when we take proteins started from the mouth, there is no digestion of protein in the mouth, and it will now pass to the stomach. So once it passes to the stomach, it stimulates the dietary protein will now stimulate the gastric mucosa to release a hormone called gastrin. And this gastrin stimulates T cells. It stimulates T cell and peritoneal cell to secrete an acid as well as the protein. So now, as we are seeing, when we take the dietary protein, this dietary protein stimulates the gastric mucosa. It stimulates the gastric mucosa to release a hormone called gastrin, and this gastrin in turn stimulates the tube cell to produce a hormone called to stimulate the production of an enzyme called pepsinogen. And this pepsinogen is an inactive enzyme. It's an inactive, which is zymogen. And then the peripheral cells will also be stimulated to produce hydrochloric acids. And these hydrochloric acids in the stomach have four major functions. Hydrochloric acid in the stomach have four major functions. And one of the functions of hydrochloric acid in the stomach, it acts as antiseptic. It acts as antiseptic. It means that whenever there is any microorganisms or any dangerous microorganisms in the stomach, this hydrochloric acid can kill them like bacteria. So the hydrochloric acid serves as antiseptic agent. It means that whenever there is any microorganisms in the stomach that can cause any harm or that can cause any damage, then the hydrochloric acid will now kill them and eliminate them. And then second, another function of hydrochloric acids in the stomach, it also acts as a denaturating agent. Denaturating agents. So generally proteins are in the three-dimensional structure. Generally proteins are in the three-dimensional structure. So it means that when you have your proteins in this three-dimensional structure, it means that all the amino acids are folded and the peptide bond itself will remain hidden. But in the presence of this hydrochloric acid, it will now denature the proteins and linearize it. So it will now linearize it. So it will now become linear and open. So that when it's now, this protein is denatured by HCl, it means that all the peptide bonds will be exposed. So that at least it will now be exposed for the digestive enzymes to recognize or to assess the peptide bond in the proteins. So the hydrochloric acid is served as a denaturating agent by denaturing the dietary proteins to form a linear chain so that the peptide bond will be accessible by the digestive enzymes. So it acts as a denaturating agents. And then the third functions of this hydrochloric acid, it helps in the absorption of vitamin B12. It helps in the absorption of vitamin B12. Vitamin B12. So this is the third functions of hydrochloric acid in the stomach. Number one, it serves as an antiseptic by killing bacteria and some dangerous microorganisms. It also acts as a denaturating agent so that it can actually linearize the proteins from its three-dimensional structure so that the peptide bond will now be accessible by the digestive enzymes. And then the fourth functions of this enzyme is the most important one. The fourth functions of this hydrochloric acid, which is the most important one, as we said here, this pepsinogen is produced as an inactive enzyme. It's produced as an inactive enzyme. So the pepsinogen produced from the tube cell are activated. This is inactive. So it is activated by HCl into pepsin, which is the active form of the enzymes. So the fourth functions of HCl is to activate pepsinogen to pepsin using the HECl itself. So it means that in this, in the fourth functions, as we said, some of the proteins in the digestion they usually produce as an inactive form. So the pepsinogen is now converted to active form using HCl. So that is the fourth function. And then at the same time, the pepsin that is produced as a result of activation of the HCl, then it now go back the pepsin itself will now go back and activate more of pepsin. It means that when you have this, you have pepsinogen. After the pepsin is being activated by HCl, then at the same time, the pepsin itself will now go further to activate more of its form. It will now go back to activate more of its form. And the process of doing this is called autocatalytic cleavage. It means that the enzyme itself is activating more of its form. So first, pepsinogen is activated to pepsin by HCl, and the active pepsinogen will now go ahead and activate more of its form from the pepsinogen to produce more of pepsin using the enzyme itself called pepsin. So this is what we call autocatalysis. We call it autocatalysis. So first, in this form, dietary protein, after you take dietary proteins, then the gastric mucosa will now be activated to release gastrin, and the gastrin is a hormone which also in turn activates tube cells to produce pepsinogen, and then to activate also peripheral cells to produce HCl. And the HCl produced in the stomach have four major functions. One, antiseptic activity, that is, it kills bacteria, and the second, it acts as a denaturing agent by linearizing the protein so that the peptide bond will be accessible. And then the third one is absorption of vitamin B12, and then the last one is the activation of pepsinogen to pepsin. And the pepsin produced will now also further activate more of its form to produce more of pepsin using what we call autocatalysis. So what this pepsin does in the stomach? This pepsin is usually cleave the protein at the amino. The pepsin here, after the pepsin is being activated, so the pepsin here acts on the amino terminal, or act on a peptide bond of amino terminal of aromatic amino acids. So it acts, or it cleaves the peptide bond at an amino terminal of aromatic amino acids. And these aromatic amino acids includes tyrosine, tryptophan, and phenylalanine. So it means that the pepsin, after it's being activated, will usually break down the peptide bond at an amino terminal of aromatic amino acids. So it means that whenever there is aromatic amino acids, the pepsin will cleave them. So pepsin generally cleave on the aromatic amino acids in the protein. After that, then it will now produce a mixture or a fragment of the amino acids that are cleaved by the pepsin. And in addition, in the stomach, we also have another enzyme which is called renin. Renin is also another enzyme that's involved in the digestion of milk in infants. So it's usually found in infants. So this enzyme renin is usually found in infants. It's usually found in infants. It's found in infants, and it also have another name called chymosin. And this enzyme renin is absent in adults. So it is found in infants, and another name for this renin is called chymosin, and it is absent in adult. So as we are actually taking milk, this enzyme is important in milk coding milk. The enzyme renin is out in coding milk. So it's found only in infants. So because as we are infant, we are taking milk. So that is why we have this enzyme renin, and it's acts on the milk. That is actually involved in milk coding, and it has another name, chymosin. It has another name called chymosin. And as we grew, because we stop taking the milk, the enzymes will become inactive. So it's absent in the stomach of the adult, because as we stop taking the milk, then the renin will be disappear. So that is the functions of this. So what this enzyme actually does is actually combats the casein of milk. The enzyme renin combats the casein in milk. So the renin combats casein in milk to grow casein, and that is the functions of renin. So renin combats the casein of milk to para-casein, and this para-casein combined with calcium ion to form what we call calcium casein. So when the para-casein and calcium combine, it will produce what we call calcium para-caseinates. Calcium para-caseinates. So this is renin. Renin is found in infant, and what it does is actually involved in coding milk, and it combats casein from milk to para-caseinate, and the para-caseinate react with calcium to form calcium para-caseinate. So ladies and gentlemen, at this point we should understand that in the stomach we have two major enzymes. These major enzymes, we have pepsin and we have renin. So pepsin is used in the digestion of proteins at the aromatic amino chain to produce a different fragment of the protein or of the peptides. And then the renin, which is also found in the mouth, is only found in infant. It is absent in adult stomach. And what it does is actually coding of milk. It converts casein of milk to para-casein, and the para-casein react with calcium to produce what we call calcium para-caseinates. So also at the same time, generally it has been known that in most cases, an individual blood group O produce a lot of hydrochloric acids. So because individual blood group O are usually produce a lot of hydrochloric acid, so that hydrochloric acid produced by the O group individual generally produce a lot of hydrochloric acid, which usually involve in activating pepsinogen to pepsin. So because there is a lot of this hydrochloric acid, so it means that even if you didn't take dietary protein, sometimes the CCR activate this pepsinogen to pepsin. And once the pepsin is activated, then it will now start searching for protein to digest. And if there is no digestion, then the pepsin will now start digesting our own protein. So it's called self-digestion. Then as a result of that, that is actually in some cases that leads to the condition of ulcer. And of course, apart from this, there is also another organism that called ulcer, also called Helicobacter pylori. But in most cases, you will see that most of all individual groups, all individual person, they have very chronic ulcer, and the reason is because they are producing so many of these HCl, which usually activate pepsin pepsinogen to pepsin, and at the end it will now undergo what we call self-digestion. So now the next thing after this, the stomach content, that is after the digestion, the peptide fragment from the stomach will now pass to the small intestine. And once it passes to the small intestine, the acidic content of the fragment triggers the expression of a hormone, and that hormone is called secretin. So now, the stomach contents, that is the product of the digestion of pepsin, will now pass to the small intestine. And once it passes to the small intestine, it triggers the secretion of hormones called secretin, which stimulate, and this secretin stimulate the production of bicarbonates. And these bicarbonates, the functions of these bicarbonates that is produced as a result of the activation or as a result of stimulation by secretin, it now actually triggers the small intestine to produce bicarbonate. And the main function of this bicarbonate is to neutralize the acidic contents of the fragments, So it neutralizes from the pH or from the lower pH, because the acidic content of the stomach is highly acidic. So this bicarbonate will now neutralize the acidic contents of the fragment and increase the pH to around 7 to 8 pH, so a pH of about 7 to 8. And then this digestion of the proteins will continue in the small intestine, and of course at the upper part of the small intestine, which is the duodenum, which also causes the secretion of another hormone called cytokinins. So it will now trigger the production of what we call the duodenum will now release a hormone called cytokinin, which trigger the secretion of some enzymes by the small intestine, and these enzymes that are in turn triggers the production by this cytokinin are trypsinogen. We have trypsinogen. We have chemotrypsinogen. We have trypsinogen, we have chemotrypsinogen, and we have procarboxyl peptidase A and B. Procarboxyl peptidase A and B. So these are the zymogens, that are the inactive form of the hormones that are produced in the small intestine, which is actually triggered by the secretion of polycythemia. And of course, this trypsinogen is the inactive form of trypsin. It's an inactive form of trypsin. And then chemotrypsinogen is also inactive form of chemotrypsin. And of course, the procarboxyl peptidase A and B is the zymogen form of carboxyl peptidase A and B. carboxypeptidase A and B. Now, the trypsinogen is activated by endopeptidases. It's activated by endopeptidases, or in some cases, you see that this endopeptidases is replaced as endokinases. So endopeptidases slash endokinases will now activate trypsinogen to trypsin. So once this trypsin is being activated, then the trypsin itself will now further activate more of another trypsinogen to trypsin. So it means that the trypsin here is also undergoing what we call autocatalytic cleavage. So trypsinogen will now activate trypsinogen will now be activated to trypsin using endopeptidases or endokinases. And the trypsin itself, the active trypsin, will now activate more of the trypsinogen to trypsin. So this is also another stage of autocatalysis. And at the same time, this trypsinogen is also responsible for activating the chemotrypsinogen pro-carboxypeptidases to chemotrypsin and carboxypeptidase A and B. So it means that in this case, as we said, first after the cholecystokinin is being activated and produced, and it will now trigger the production of chemotrypsinogen, pro-carboxypeptidase A and B, and then the trypsinogen is now activated using endopeptidases and endokinases. So when it is activated, the trypsin or the active form of the trypsinogen, which is trypsin, will now activate more of the trypsinogen to trypsin. And at the same time, the same trypsin will now also activate the chemotrypsin. It will now activate the chemotrypsin as well as it will activate the chemotrypsinogen pro-carboxypeptidase A and B. So it means that in this case, the key player is trypsin. So trypsin is the one that is responsible for activating all enzymes in the small intestine. Okay, so what this trypsin does is actually it's an enzyme that cleave on. So trypsin is an enzyme that cleave protein at arginine and lysine. So this is an enzyme that cleaved the polypeptide chain at arginine and lysine polypeptide chain. So whenever there is arginine and there is lysine, the trypsin will now cleave and break down the protein. So, and then the main functions of chemotrypsin in the small intestine is also cleave the carboxylic group of aromatic amino acid. But remember, we've seen it cleaved amino group of aromatic amino acid. But chemotrypsin, it cleaved carboxylic group of aromatic amino acids. And then, of course, apart from the chemotrypsin, which is cleaved the aromatic amino acid and the carboxylic group, it cleaved aromatic amino acids, but at carboxylic group. And then, so the next one, and we also have, in addition to trypsinogen, chemotrypsinogen, pro-carboxypeptidase, there is also proelastase And of course, this pro-elastase also activated to elastase, which is the active form. And of course, the activation is still using trypsin. So trypsin is a key player that also activates pro-elastase to elastase. And of course, this elastase is an enzyme that also cleaves the amino acids, like it cleaves the amino acid like glycine, it cleaves amino acid like glycine, serine, it cleaves amino acid like glycine, serine, and alanine. So this is the functions of pro-elastasis is also activated to elastase using trypsin, and the elastase is actually involved in cleaving amino acids like glycine, serine, and alanine. So then the next thing after this one is carboxylpeptidase A and B. So what carboxylpeptidase A and B does? So then carboxylpeptidase is a zinc-containing enzyme. It means that it's an enzyme containing zinc as a core factor. And of course, as we said previously, this carboxylpeptidase is activated by trypsin. And of course, as we said, we have carboxylpeptidase A and B. So carboxylpeptidase A is an enzyme that cleaved amino acid. It cleaved amino acids at a carboxyl terminal peptide bond. It cleaved amino acids at carboxyl terminal peptide bond connected to trypsin, phenylalanine, and tyrosine. So it means that it's an amino acid that cleaved the peptide bond of aromatic amino acid at carboxyl terminal. It cleaved aromatic amino acids of peptide bonds, or it cleaved the peptide bond at carboxyl terminal. So it cleaved aromatic amino acids. And then the carboxylpeptidase B is also another another enzyme which actually cleaved at a carboxyl terminal bond connected to the arginine and glycine. So this one at a carboxyl terminal of aromatic amino acids, it cleaves aromatic amino acid at carboxyl terminal. And then for the carboxylpeptidase B, it cleaves amino acids such as arginine and glycine. Arginine and glycine. Also at carboxyl terminal. Cleave the peptide bond. It cleaves the peptide bond of arginine and lysine at carboxyl terminal. That is carboxylpeptidase A and B. So this is the role of carboxylpeptidase. So, and then the next enzymes that we are going to look at here is, so the next enzyme is aminopeptidase, So aminopeptidase is an enzyme that cleaves amino acid at an amino acid terminal. This one is carboxyl terminal and this one is amino terminal. And there is also a class of enzyme which is di and peptidase. Peptidase. It means that if there is a peptide that contains only two amino acids, then the dipeptidase will now cleave on it. And then tripeptidase will now cleave on three peptides. So all this thing is to ensure that the proteins is completely break down to produce individual amino acids. So amylopeptidase is to act on the amino acid at the terminal, at an amino acid terminal, and then di and tripeptidase is to cleave dipeptide and tripeptides into the individual amino acids. So you see, ladies and gentlemen, the digestion and the absorption of protein. The digestion actually starts from the stomach by pepsin and renin, and renin is only found in an infant, and then the pepsin is an enzyme that acts on the aromatic amino acids, and then it's now passed to the small intestine where bicarbonate is being produced to neutralize the content. So that is how it follows. So the aim of this digestion of all these enzymes is to produce free amino acid, which can be absorbed. So the next thing now is the absorption of amino acids.